Hello students, welcome back to my channel. In this video, I am going to give you a brief lecture on C4 cycle. Okay, in the previous video, we have already discussed about C3 cycle. So, in case of C3 cycle, there is an enzyme ribosco, ribulose bisphosphate carboxylase oxygenase. So, which helps in the carboxylation. So, it acts as a carboxylase. So, it adds carbon dioxide molecules to the RUBP. Rubisco acts as a carboxylase. So, it adds CO2 to the RUBP, results in the formation of 3-PZA, phos-3-phosphoglyceric acid. Okay, so 3-PZA undergoes sequential reactions, results in the formation of sugars. In this way, in the dark reaction, sugars are synthesized. But in case of C4 plants, nothing but plants are living in tropical and subtropical regions, there the conditions are different. So there the conditions like high light intensity, high oxygen concentration, high temperatures are present. During these conditions, so RUBP acts as a oxygenase. So it shows the oxygenase activity. So due to the rubisco is having a dual nature. So due to the dual nature of rubisco, in the conditions like high light intensity, high O2 concentration, high temperatures, it acts as an oxygenase. It shows the oxygenase activity. Then what happens? So it adds O2 to the RUBP instead of carbon dioxide. It shows the oxygenase activity. Then what happens? So 1 PZA molecule and 1 glycolate. 1 glycolate molecules are found. Okay. So glycolic acid or glycolate is formed. So when the conditions are like this, so instead of adding carbon dioxide, rubisco adds oxygen to the RUBP. So then what happens? Instead of 2 molecules of 3-PZA, 1 molecule of PZA is formed. Okay, and 1 molecule of glycolate is formed. Actually, it is a wasteful process. Instead of fixing energy, it consumes energy. So, the plants are living in tropical and subtropical regions. They are performing additional cycle, nothing but C4 cycle to avoid the photorespiration loss. So, to avoid the photorespiration loss, so plants are living in tropical and subtropical regions. They are performing additional cycle to avoid the photorespiration loss. Okay, so let's see the C4 pathway. C4 cycle was first discovered by the two scientists Hatch and Slack. They discovered a C4 cycle in sugarcane. That's why this cycle is also called as Hatch-Slack pathway. So this cycle is also have another two alternative names such as beta carboxylation pathway and cooperative photosynthesis. of C3 plants, 3PZA is the first stable compound. So which is a 3-carbon compound. That's why that cycle is called as C3 cycle. Whereas in case of C4 plants, so the first stable compound is oxaloacetic acid, which is a 4-carbon compound. That's why the cycle is called as C4 cycle. That's why the cycle is called as C4 cycle. This cycle is operated in many species of Graminaceae like maize, sorghum, sugarcane, etc. It has also been found in Atriplex and Chenopodium too. Okay? Actually, C4 plants are evolved to overcome the photorespiration loss. Okay? So, they evolved to avoid the photorespiration. Okay? So, these plants exhibit two important features such as absence of photorespiration. Second one is Kranz anatomy. How they avoiding the photorespiration loss, let's see later. So let's see the Kranz anatomy first. So Kranz anatomy is nothing but C4 plants are having two types of photosynthetic cells. Two types of photosynthetic cells. Such as bundle sheath cells and mesophyll cells. So these leaves are called as dimorphic leaves. That means the leaves of C4 plants are having two types of cells. They are nothing but mesophyll cells and bundle sheath cells. Okay. So let's see what are bundle sheath cells, how they are performing C4 cycle. So C4 plants are showing the Kranz anatomy. Kranz anatomy is nothing but plants are having two types of photosynthetic cells. Nothing but mesophyll cells and bundle sheath cells. So have a look. In case of these are the bundle sheath cells. This is the bundle sheath. This is the bundle sheath cell which is surrounding the stem. These are the mesophyll cells. Okay. So this is a dimorphic leaf present in C4 plants. Dimorphic is nothing but two types of cells are present. So they are nothing but mesophyll cells and bundle sheath cells. Bundle sheath is surrounding the stem. Okay. So let's see the differences between bundle sheath cell and mesophyll cell. So if you see the mesophyll cell, it consists of chloroplast having grana chloroplast. If you see the chloroplast of mesophyll cell, that is consisting grana. Whereas in case of bundle sheath cell, there is no grana, there is only presence of stroma lamellae. There is only stroma lamellae. Okay. So this is the difference between mesophyll cell and bundle sheath cell. So mesophyll cells are having granule chloroplast, whereas bundle sheath cells are having agranule chloroplast. You can see mesophyll cells are having smaller chloroplasts. The cells are also small and the chloroplasts are also small. Whereas the bundle sheath cells are bigger than when compared to the mesophyll cells, and the chloroplasts of bundle sheath cells are also bigger. Okay. So Kranz anatomy means wreath or ring. Bundle sheath cells are surrounding the steel in a ring-like, in a wreath-like manner. So that's why this anatomy is called as Kranz anatomy. Okay. So let's see the C4 cycle. So this is the actual C4 cycle. So occurs in C4 plants are having two types of photosynthetic cells, nothing but mesophyll cells and bundle sheath cells. Okay. In case of C4 plants, CO2 is accepted by PEP, not by RUBP. Okay. So results in the formation of oxaloacetic acid in presence of an enzyme pep carboxylase. Okay. So, oxaloacetic acid is a 4-carbon compound. That's why this cycle is named as C4 cycle. Okay. After that, the oxaloacetic acid converts into malic acid or aspartic acid, which is a 4-carbon compound. This malic acid is diffuses into the bundle sheath cell. Bundle sheath cells are present around the steel. Okay, in a wreath manner, in a ring-like manner. Okay, then the malic acid undergoes decarboxylation. Okay, so then it converts into pyruvic acid. So this carbon, so this released carbon in bundle sheath cell that undergoes Calvin cycle. Here glucose formation is occurs. So where do we find the glucose formation? In case of bundle sheath cell, not in the mesophyll cell. Okay. So next one, the pyruvic acid again enters into the mesophyll cell. There it is regenerated into PEP, phosphoenol pyruvic acid, with the utilization of ATP. It occurs in the mesophyll cell. Okay. Here you have to observe one thing. During malic acid conversion into pyruvic acid, CO2 molecules are accumulated in the bundle sheath cell. So the CO2 accumulation is important for the C3 cycle. That's why the PCR cycle is going on here. Okay. So next one, when compared to mesophyll cells, mesophyll cells are located towards the upper epidermis, whereas the bundle sheath cells are present little inner side. Okay. So that's why here the temperatures are low when compared to the mesophyll cell. In this way, they are avoiding high temperatures. So, carbon accumulation, first one, carbon accumulation occurs in the bundle sheath cells that facilitates the PCR cycle or C3 cycle, and they avoiding high temperatures by changing the site of photosynthesis. Okay, oxygen is evolved due to the light reaction only. The light reaction performed in mesophyll cells, not in bundle sheath cells, because mesophyll cell chloroplasts are having granule chloroplasts. They are only able to perform the light reaction. That's why the oxygen concentration in the bundle sheath cells is low. So in this way, they are avoiding photorespiration loss. So let's see these equations in detail. Okay. So these are the steps occur in C4 cycle. In the first step, so that is carboxylation step. So PEP accepts carbon dioxide in presence of an enzyme pepco. Phosphoenol pyruvic acid is going to accept CO2 molecules in presence of an enzyme pepco, results in the formation of 4-carbon compound, that is oxaloacetic acid. In the second step, oxaloacetic acid undergoes reduction reaction. Okay, second step is the reduction reaction. Here it undergoes reduction, so it reduces into malic acid. Here 1 NADPH is used for this reaction. Second step is the reduction reaction. Here you have to know one thing. Some species are synthesizing malic acid, whereas some species are synthesizing aspartic acid. So if malic acid is synthesized in the C4 cycle, those are called as malate formers. If aspartic acid is synthesized in the cycle, they are called as aspartic formers. Okay, so for example, sorghum, maize, they are synthesizing malic acid, whereas at triplex, they are synthesizing aspartic acid instead of malic acid. So the synthesized malic acid in mesophyll cells, they get transported into the bundle sheath cells. In the bundle sheath cell, the first two steps are occur in mesophyll cells, whereas the third step occur in bundle sheath cell. In the third step, either malic acid or aspartic acid, it undergoes again oxidation reaction. It undergoes oxidation reaction, it results in the formation of pyruvic acid. Okay, so here in this step, one CO2 molecule is involved in the bundle sheath cell. Okay, so in this way CO2 is accumulated in the bundle sheath cells that facilitates the C3 cycle. That facilitates the C3 cycle or PCR cycle. Okay, so let's see the next step. Pyruvic acid enters into the mesophyll cell for the regeneration of PEP. There, in presence of an enzyme dikinase, it undergoes phosphorylation reaction. Here, the two ATPs are utilized. Okay, so one pyruvic acid is regenerated into again PEP, phosphoenol pyruvic acid. Okay, so these are the steps occur in C4 cycle. In case of first reaction, phosphoenol pyruvic acid carboxylase that catalyzes the reaction. In the second step, malic dehydrogenase catalyzing the reaction. In the third step, malic enzyme catalyzing the reaction. In the fourth step, pyruvic acid dikinase that catalyzing the reaction. So let's see the energetics of C4 cycle. For the fixation of each CO2 molecule, 2 ATPs are required additionally. So for the fixation of 6 CO2, for one molecule of glucose synthesis, 6 carbons has to fixed. Okay, for 6 CO2, 2 into 6, that is equal to 12 ATPs are required. For the C3 cycle, it is spending 18 ATPs. 18 ATPs are required for the performance of C3 cycle. Okay, so totally 30 ATPs are required by C4 plants for the fixation of one glucose molecule. By spending 12 ATPs additionally, they are overcoming photorespiration. This is about C4 cycle, students.